So hi everyone. So our research is about optimizing Arthrospira platensis, which is a type of cyanobacteria. So it's growth for biofuel production. So here's our table of contents. We're going to first go over our background and hypothesis. Then we'll talk about our procedures and then go into like observations. And then lastly, we'll go over like results and our analysis, limitations and future steps and research that we're going to do further. Okay, so anthropogenic activities, which involve the release of greenhouse gases, have caused climate change. And as climate change is exacerbated, the need for sustainable solutions, such as like bioenergy, has grown. So as this interest or exploration of bioenergy has grown, the interest in cyanobacteria has grown as well. And this is because cyanobacteria has been identified as a solution for the production of generation biofuel. So third generation biofuel would be using microorganisms to produce biofuel, whereas first generation would be like using crops like wheat. So cyanobacteria, we didn't switch the slide, sorry. So cyanobacteria works in this way because it produces compounds such as hydrocarbons that can be utilized as precursors of biofuel. So it's a lot more efficient than previous methods of biofuel production. And a significant like benefit of using cyanobacteria is that it does not interfere with the agricultural industry, whereas like wheat would. So it can like use very limited land, and in favorable conditions, biofixation efficiency of cyanobacteria can be about like tenfold more than that of terrestrial plants. And then cyanobacteria also exhibits the capacity to produce lipids at rates like 100 times or more than conventional plants. And this is significant because we need the lipids to produce the biofuel. And cyanobacteria even outperforms algae, which is also a current alternative. So out of many cyanobacteria species, Arthrospira platensis is especially attractive because of its high growth rate and also because of its high amounts of lipids in its thylakoids. And then lastly, also because it's most used and most available at the moment. So in our research, we also decided to work with two other species, so Nostoc and Anabaena species. So Nostoc species, they live in large colonies and they live in these kind of envelopes. And then whereas Anabaena, they kind of live in like free clusters or they kind of clump up into macroscopic mats. So both of these species have or form symbiotic relationships with other organisms in their natural habitats. And this is mainly because of their ability to fix nitrogen. So both of these species have unique cells called or named heterocysts, and they basically allow the species to fix atmospheric nitrogen and turn it into ammonia and distribute it throughout its environment. So in, for instance, like Nostoc's natural environment, it can create these symbiotic relationships with other types of bacteria and plants such as water ferns and mosses. It provides nutrients to these plants, aiding in the plant's growth, while the plants, for instance, can offer like a habitat or other types of nutrients for the cyanobacteria. And through research, we have identified that Arthrospira platensis growth increases when additional sources of nutrients are added. And thus, next slide, please. And so our hypothesis is that due to Anabaena and Nostoc species' abilities to fix nitrogen, we hypothesize that they will improve environmental conditions when being co-cultured with our target species, Arthrospira platensis, and allow for improved growth as compared to when Arthrospira platensis is grown by itself. Ahana, I think it's your slide.
Sorry about that. So we started by using a water pump for Arthrospira, but we stopped because the pump was pushing out too much water too forcefully, which was harmful for the species. Instead, we switched to stirring the Arthrospira by hand for 10 minutes each day. Hand stirring helped the Arthrospira grow quickly. We stirred it every day except for one day. When we didn't stir, the species remained healthy. However, we didn't stir for 2 consecutive days. There was a noticeable difference in the growth. Next slide, please. As we can see in the picture above, these are the non-homogeneous mixtures. These pictures were taken before the mixing. Nostoc is settled in the bottom. We can see that it's fuzzy. Anabaena is most likely settled at the bottom, but there are a couple clumps that are surfaced at the top, as well as there are small bubbles on the surface. On the previous slide, we saw that there were non-homogeneous mixtures, so we made it into a homogeneous mixtures. Under the microscope, we observed specific characteristics of different algae species. Arthrospira exhibits a distinct blue-green coloration and a spiral shape. Nostoc appears as bead-like chains that form clusters, also displaying a blue-green hue typical of algae. Anabaena, similarly, a blue-green algae represents in clumps.
So firstly, for our procedure, all materials were sterilized using an autoclave, and we used a Faceta LED growth light, along with 6 autoclave blue flasks. Next, we prepared the stock solution, maintaining a ratio of 10 ml of stock to 200 ml of media. The control group consisted of 100 ml of media along with 10 ml of A. platensis. In the independent trials, we used 200 ml of media for each setup. Both independents had 5 ml of A. platensis and 5 ml of their species, which was either Nostoc or Anabaena. The control and both independent trials were stirred for 10 minutes daily for 33 days. This was our setup of 2 trials. Starting from the left, we have the control, Anabaena, and the Nostoc from trial 1, and then on the right, we have the same for trial 2. These were our cultures on the day we began, and they all started with about the same amount of growth. The data we collected are the microscope photo comparisons, the light absorbance, which we measured using the spectrophotometer, and the daily observations on the cultures. These are the recorded absorbance values at 600 nanometers for each sample. We observed significantly higher values in samples with A. platensis combined with Anabaena compared to the controls and the other combinations. This was on the 6th day of observation. We can see some growth in all of the cultures. This was on the 10th day of observation. We can see that the Anabaena culture has slightly more growth than the other 2. Comparing the trials on the 10th day, we can see that the cultures have similar amounts of growth.
And then so here is our 2-week mark. So on this day, we had done the measurements all again to compare to our first day. Through many of the papers that we've read, a lot of these cultures go on until 2 weeks. So we decided to do that as well. And later after 2 weeks, we did continue like observations. So at this 2-week mark, we noticed that the colors of Arthrospira Platensis and Anabaena species, those cultures, and Arthrospira Platensis and Nostoc species culture became increasingly similar, which is a difference that we did not observe prior to this. And then on to the next slide. So here on the next slide, we measured with the spectrophotometer again, and we did notice that the Nostoc co-culture did increase in light absorbance quite a lot, but then also still all the co-cultures were more dense than our control cultures. And then here we also used the microscope to get a closer look. So actually when we were like making observations for the species when we were mixing before the 2-week mark, we noticed also that there was a lot of clumping, especially in our control group. So there was a lot of clumping even when we tried to make it into, even when we were mixing, you can still see like some visible clumps. And then so under the microscope, you can see that our control variable, and this is in our first trial, it was completely like overwhelmed with green algae cells, which is we identified as Chlorella vulgaris, whereas the other two, the co-culture with Anabaena species and co-culture with Nostoc species didn't see that. And then so we identified that it was because of these like invasive cells that grew, that there was a lot of clumping, especially in our control group, whereas that wasn't observed for the other cultures. And then here we have our second trial, which also observed a similar pattern. And then we noticed that the Anabaena cultures had probably the most amounts of spirulina growth.
This is day 20 of the species. As you can see, they are very healthy. They have like a very green, vibrant color. Here are the pictures of the species before mixing. The colors are vibrant, and if you look closer, there are like more bubbles at the surface. These are the pictures of the species before mixing. As the days go by, like the color has been becoming more dull and there has been less bubbles coming up at the surface. Here are the species underneath the microscope. The top row are like more closer pictures and the bottom row are like further away. This is the trial 2 of the species. The top row are the more closer pictures, and then the bottom row are the farther away.
And so before I move on to this slide, I also wanted to mention that on day like 33 and like towards the end, we noticed that there are less and less Arthrospira platensis cells. So that was the same across all of our cultures. If we go to the slide before, we can see that it's lacking those spiral-looking cells, which are the Arthrospira platensis. That just kind of confirms or shows us that there was like competition with the nutrients since it was like a long time since we had added the new media from the very beginning of the experiment. So that's something I wanted to note. And then, so on the next slide, where we have the spectrophotometer measurements, we still kind of see that the control groups were like least dense, whereas the Arthrospira platensis and Anabaena cultures, as well as Arthrospira platensis and Nostoc species cultures had the higher light absorbance. And then what was interesting was that towards the end, the cultures with Nostoc species seemed like they had the most light absorbance. And then so after we had kind of compiled all of our observations and data, we conclude that our research, which is still ongoing, but our research so far supports our hypothesis. And just mentioning again, our hypothesis was that since Anabaena and Nostoc species are able to fix nitrogen, and due to their properties, they will be able to improve environmental conditions and allow for improved growth for our target species, which was Arthrospira platensis. And this would be better than if we grew Arthrospira platensis by itself. So through what we observed, we noticed, or we're still on the previous slide, sorry. Through what we observed, we still noticed that when we co-culture Arthrospira platensis, especially in this very nutrient-rich environment, it lowers the chance when we co-culture, it lowers the chances of algae growth, which would be the Chlorella vulgaris growth. And this is significant because in the controls, we saw that there was a lot of clumping, and even when we mixed it, we could still see there were some pieces that were really clumping, and even under the microscope, we could see that like intense amount of Chlorella vulgaris. And so this is significant because clumping inhibits growth of our target species, and that is actually the whole reason why, like this inhibiting of growth when clumping is the whole reason why Arthrospira platensis can't live in stagnant waters, and that's why we have to stir every day. And lastly, through this extended period, after the 2-week mark, we can kind of just see like overall, especially with the Anabaena co-culturing, there was like the most amounts or the healthiest looking Arthrospira platensis. So for our limitations, it was that the non-controls, which is our co-cultures, had more media. So in order to maintain the ratio for it all to be the same, like stock culture to media, we had to keep our controls to 100 milliliters, whereas our co-cultures had 200 milliliters. And then, so that was one limitation. And then another limitation was that we had like the places that we had gotten the species, we couldn't find the very like specific strain. And then another limitation would be like sample size. Next time we could test with larger samples, space limitation, and then also better environment. And then on the next slide, we have future directions. So currently we're still observing the species, and we're still kind of determining what to do next. But one of the future steps that we're considering is setting up another experiment with different media ratios. For instance, we could consider like lowering the nutrient amount and also adding in some distilled water. And then another possible thing we can consider is to test with other species that are possibly faster growing than Arthrospira platensis. And then lastly, we would like to thank our advisor, Dr. Kaur, for her continuing support and guidance throughout our work.